Hello students, myself RK Mishra. Now we are going to discuss about the functional property of EcoR first. In previous video lecture, I have aware you the EcoR first is a type of restriction endonuclease enzyme, and more detail about restriction endonuclease enzyme was you can study in previous video lecture. With the help of that video lecture, you will also aware yourself what are the different strategies that make restriction endonuclease differ from exonuclease. Listen, there is need to know what are the strategy plan. Now back to more detail about restriction endonuclease, and among the restriction endonuclease there is EcoR first. In your syllabus in class 12, EcoR first and their action is more important. Mechanism of action of EcoR first are actually in your syllabus. So before knowledge about the action of EcoR first, there is need to know how EcoR first act over the DNA molecules and what are their different sequence that selected by EcoR first. So let us talk about the functional property of EcoR first. In previous video lecture, you have learned the different kinds of restriction endonucleases and their nomenclature, in which the first letter, that is E assigned for the genus of the source organisms. The second two letter is actually the first two letter of species. The second last letter represents the strain by which the EcoR first or any other kinds of restriction endonuclease get selected, and the first, second, third means the last Roman digits always represent the order of isolation. There are about 900 restriction endonuclease still discovered from 213 strain of different bacteria that prokaryote mostly is able to generate the restriction endonuclease. So overall 900 restriction enzyme, more than 900 restriction enzyme was actually discovered from 213 strain of bacteria. So what are the strain, and if you have any doubt regarding the strain, you can frequently ask the previous video lecture. You can frequently learn the previous video lecture whatever the important strain, what are the concept of strain. Now in this video lecture, let us talk about the functional property of EcoR first. Actually EcoR first select the specific recognition sequence on DNA. Specific recognition sequence on DNA. The specific recognition sequence selected by EcoR first called palindromic sequence. Listen, and the palindromic sequence can be defined as the plane mirror opposite nitrogenous base pair sequence on the different strain of DNA. Listen, क्या होता है plane mirror opposite? First of all, there is need to know the palindromic sequence. पहले हम palindromic sequence को बता दें कि ये टर्म क्या होता है. ये टर्म पहली बार आपके लाइफ में आया है, तो पहले हम palindromic sequence को समझा दें. palindrome क्या होता है इसके बारे में जान लें. palindromic sequence क्या होता है, पहले हम इसके बारे में समझ लें. ठीक है, फिर हम समझेंगे कि इसका रोल क्या होता है. ठीक है, सबसे पहले हम palindromic sequence समझेंगे. The plane mirror opposite base pair sequence. The plane mirror opposite base pair sequence on DNA strand called the palindromic sequence. called the palindromic sequence. I hope your concept from this definition is almost clear. The plane mirror opposite base pair sequence means, as you all know, on DNA strand, suppose that this is the one strand, polarity 5' to 3', and this is another strand, polarity 3' to 5'. Suppose that one strand has nitrogenous base pair sequence, then opposite, that is the second strand, will have the opposite but plane mirror sequence. As like the word Malayalam. Suppose that I am going to write about the word Malayalam, M-A-L-A-Y-A-M. It means either you start reading from the right side or from the left side, there will be the similar spelling you will got. For this definition, for this word you will got the similar kinds of spelling. Malayalam, and from the right side there will be the Malayalam. It means this kinds of sequence actually should available on the polarity of first strand and polarity of second strand, that is on DNA first and second strand. So which kinds of DNA sequence is actually selected by the ECoRI first? The palindromic sequence. And which kinds of palindromic sequence? Listen carefully. The ECoRI first select the GAATTC. Palindromic sequence GAATTC. In the opposite direction, the complementary of C will be G, complementary of T will be A, the complementary of T will be A, complementary of A will be T, complementary of A will be T, and complementary of G will be C. It means when you start reading from the 5' polarity, you will get GAATTC. And also, if you are going to start this sequence on the second strand, that is the polarity 3' to 5' from opposite side, there will be the similar term, the GAATTC. It means GAATTC from the 5' to 3' polarity will be opposite of the polarity 3' to 5'. It means these two are actually plane mirror opposite sequence. This plane mirror opposite sequence are actually called as palindromic sequence. Palindromic sequence, and this sequence is actually selected by ECoRI first. So ECoRI first actually select this kinds of sequence after, for the cutting to the during the process of recombinant DNA technology. So this is the specific features, that is which kinds of sequence selected by the ECoRI first restriction enzyme. Now back to the mechanism of action of ECoRI first. This is the functional properties of ECoRI first. Now back to study the functional property of ECoRI first. You make the screenshot for this in order to note down the important concept. Now back to erase the overall data in order to study the mechanism of action of ECoRI first. Mechanism of action of ECoRI first. ECoRI first. Listen. Mechanism of action of ECoRI first. This is very important terminology in order to understand how do ECoRI first works. So first of all, there is need to learn which kinds of sequence actually selected by the ECoRI first. The restriction sites GAATTC. Suppose that this is the plasmid DNA. The portion of plasmid DNA having polarity 5' to 3' and 3' to 5'. And this is the source DNA. Source DNA or the desirable DNA, polarity 5' to 3' and 3' to 5'. Now let us see which kinds of sequence selected by ECoRI first. The GAATTC from the plasmid and also from the source DNA. Listen. So on the plasmid there will be GAATT and C on the one strand and GAATTC on the other strand. C Similarly, same kinds of sequence should be required even if you want to go the cutting of both plasmid as well as the recombinant DNA. So this is the source DNA, and this kinds of DNA is actually have the similar kinds of the sequence, that is recognition sequence or palindromic sequence. So this should have the G double A double T and C on the one strand, and on the other strand G double A double T and C on the other strand. So we can say that there should have the similar kinds of palindromic sequence both on the plasmid DNA as well as the source DNA. That is the basic point for restriction endonuclease enzyme EcoR first. So EcoR first, if do not found the similar kinds of palindromic sequence either from the source or the plasmid DNA, then EcoR first left the idea of cutting. यानी EcoR first को अगर ऐसा palindromic sequence जो फर्स्ट में है सेकंड DNA में नहीं है तो अपने कटिंग की आइडिया को पूरी तरह से एलिमिनेट कर देगा, काटेगा ही, ठीक है. बट हियर आई हैव ड्रॉन द सिमिलर काइंड्स ऑफ सीक्वेंस फ्रॉम द सोर्स DNA as well as the plasmid DNA. Listen, so let us talk about which kinds of portion in between the G double A double T C from the first or second strand gets selected by the EcoR first. Listen, actually EcoR first cut the guanine and adenine, cut the strand between the guanine and adenine on the DNA. It means it left at the middle portion, that is cut the DNA just away from the middle of G double A double T C. This is the overall palindromic sequence. This is the palindromic sequence, and this is the middle of palindromic sequence. Actually, the EcoR first. This is the EcoR first. This select the portion just away from the palindromic sequence toward the guanine and adenine side. So, one portion of on plasmid DNA is selected by the EcoR first as GA on 5' polarity towards 5' polarity, and the GA on the second strand also toward the 5' polarity. And similar kinds of palindromic sequence in between the guanine and adenine are also selected by the EcoR first on the source DNA. So this is the source DNA. Guanine adenine selected by the source DNA from the at the polarity 5' to 3', and guanine adenine also selected between on the source of DNA between the 3' to 5' polarity. It means what we learned from the selection of EcoR first, the EcoR first do not select the midpoint of palindromic sequence. It select away from the palindromic sequence. It select the point just away from the palindromic sequence and in between the guanine and adenine. The characteristics of EcoR first, it's always select the nitrogenous base sequence between the guanine and adenine. It means we should learn between guanine, adenine and guanine. This will be selected by EcoR first, whether they are palindromic sequence on the source DNA or palindromic sequence on the vector DNA or plasmid DNA. Listen, now back to what happened after cutting. If this will cut, then suppose that the remaining portion are there, the remaining portion of DNA are there, and the remaining portion of this DNA will be there. Then let us talk about what happened for this DNA and what happened for this DNA. Listen, the portion of this portion is actually get eliminated, and only this portion will get eliminated. So let me draw the structure after getting loss of these fragments. Listen, after application of EcoR first, only G remain on this strand. Only G remain, and CTTAA will remain on this strand. So what we learn? What we left after the cutting? Listen, only G remain and rest AATTC will be eliminated, and CTTA will be remain in the second strand. The CTTA and A will remain on the second strand, and there will be the triple bond, and this is the double bond. That will be the part of plasmid DNA. Now back to what left after the cutting, after the application of the EcoR first on the this kinds of DNA, that is the source DNA. Listen, this is the source DNA, and this portion only get eliminated, while this whole portion will get eliminated after the application of EcoR first enzyme. So what left after the cutting? Listen, adenine, thymine, and cytosine will remain in this strand, and guanine having triple bond will remain on this strand, while cytosine, thymine, adenine will get eliminated, and this will also have the double bond. So this is the sticky end. This is the sticky end. That is the specific features of restriction endonuclease enzyme. This is also the sticky end. That is the specific features of restriction endonuclease enzyme. When we join these two strands, there will be a ligation. When we join these two strands, there the process is known as ligation. So what happened after the ligation? Listen carefully. When you ligate this DNA, then you will got the hybrid DNA, guanine having triple bond with the cytosine, and thymine, adenine will remain as usual having the double bond, and this will suppose that link with it. It means adenine, thymine, adenine, thymine having the double bond, and cytosine guanine will remain as usual. Cytosine and guanine remain as usual, while the strand is actually from the second or source DNA. So we can say that this kinds of hybrid DNA is actually needed to create during the process of biotechnology, and this is very essential process. Listen, without this essential process, we cannot learn whatever the importance of recombinant DNA. This hybrid DNA is known as recombinant DNA. This hybrid DNA is known as recombinant DNA, and recombinant DNA is also known as hybrid DNA. Hybrid DNA. So what we learn from hybrid DNA? The DNA having information of two different sources. The source from plasmid and source of the main DNA, that is the source DNA. So this is the source DNA, this is the plasmid DNA, and that overall DNA is the portion of recombinant DNA. I hope your concept for recombinant DNA and the function of ECoRI first is clear. ठीक है, अब आप इसका स्क्रीनशॉट ले लीजिए. Very good. Then this need to learn by you in your own language. Thank you.